Biologists, it's week 4, and this is video 1 on understanding diffusion. Last week you downloaded some software to be able to do statistical analyses, and we also asked you to download an extra set of software called Logger Pro that's going to be used this week for collecting data on our diffusion experiments. If you haven't downloaded this already, please make sure that before you get to lab, you click on one of these links in the PowerPoint video or in the software to download link from last week's module so that you can get a copy of Logger Pro from Vernier. This is a software program that we can use to collect data using some electronic probes, and then we can save that data directly to your computer for further analysis. At least one computer per lab group is going to need this software in order to complete the lab this week. In addition to downloading software and having it installed on your computer, there are 2 questions that are dedicated to making solutions that are at the beginning of your pre-lab. So please make sure that you download the lab early from the week 4 module. After you watch this video would be great, and work through those 2 problems. They should be familiar concepts to you from chemistry and biology classes because they deal with making solutions and molecular weights of compounds and dealing with the idea of molarity for concentration purposes. Please make sure that you complete these 2 problems and you bring them into lab because they're going to be used as an entrance ticket and as an introduction to discussion as we move into the experiment. There will also be some reading in the required textbook, A Student Handbook for Writing in Biology by Karen Knisley. You had the option of purchasing either the 4th edition or the 5th edition. Both of them have mostly the same information. But what I'd like you to read are the passages on graphing data, presenting data in a tabular format, and producing figure legends for images that would serve as the data results in a lab report. The page numbers are going to differ depending on which edition you look at. 57 to 75 in the 4th edition, or 59 to 73 in the 5th edition. And this is going to serve you well because after this week's lab, you will have collected some data that's going to be used to write a rudimentary lab report that's going to be turned in 2 weeks. And we'll talk about that in another video. The concept that we're going to be focusing on this week is diffusion, which is critically important in biological systems. And after this week's lab and pre-labs, you should be able to answer the following questions to help you understand the material in the lab. Video 1 that you're currently watching is going to help you answer the question, what is diffusion, and what sort of environmental factors or cellular factors can affect the rate of diffusion. Video number 2 will help you answer the question, how do I understand dilutions, and what does dilution have to do with diffusion? Then finally, video number 3 is going to help us understand how we'll calculate some of the rates from this week's lab, or how do I calculate a rate from the slope of a line using linear regressions. I like to use somewhat silly examples to describe somewhat complex ideas. So let's look at an example using one of those magic trees that you would hang on your rear view mirror in your car because your car smells really bad. Let's also imagine that we are all currently together sitting in a large lecture hall, and I'm standing at the front, and I hang one of these magic trees from the whiteboard right behind me. Now fairly quickly, as soon as I open up the package and take this magic tree out of the plastic, I'm going to start smelling this forest fresh scent. People sitting in the front row a little bit later are going to start smelling this because the aroma of my magic tree is spreading throughout the room fairly slowly, and it might not be until the end of the class when the room starts smelling the aroma from this magic tree. But eventually the entire room will have a uniform aroma of magic tree pine forest fresh scent, which helps us understand diffusion because diffusion is the tendency for small molecules or any molecule to spread out evenly into the available space that it's given. In this case, the molecule that we are interested in are the scent molecules that carry the aroma of our pine tree throughout the entire classroom. And at the front of the room, right next to the source, those molecules are at very, very high concentration. And as soon as I take it out of the package, they start moving throughout the room through this process of diffusion. So from our classroom example, we can help explain how substances diffuse down their concentration gradient. And we can define this gradient as being a slope where in one end we have molecules at high concentration. In our slide here, our example would be these yellow balls, which are at high concentration at one side and at a concentration of 0 or very low concentration on the other side of our little barrier right here. Purple balls we could consider independently. They are at high concentration on the right-hand side and a low concentration on the other. And because substances diffuse down their concentration gradient, they're going to move from high to low concentration. Because molecules and atoms are constantly in motion, they're going to bounce against one another, and that bouncing is going to cause them to ricochet and spread out around their available space. So the yellow balls are going to start bouncing, and they'll spread out until they make it to the right-hand side of our barrier. And the purple balls are going to do the same thing. They're going to bounce against other molecules, and they'll generally start to spread out, just like when you hit a racked set of pool balls with the cue ball. One ball hits them, the rest all bounce off in different directions. So we can see that there's a net diffusion of yellow balls over to the other side and a net diffusion of purple balls to the left side until they reach equilibrium, where the concentration throughout our entire system of either color ball is generally equal. Now the movement doesn't stop here because these balls are still colliding against one another. They're still moving from one compartment to the next through the pores in our barrier. But the net movement doesn't change. We have reached equilibrium where everything stays mostly equivalent. This is also a great demonstration of how solutes diffuse independently of each other. The yellow balls on our left-hand side don't care about the concentration of the purple ball. Also, please recognize that I'm giving some sort of agency to these independent balls over here, as if they have a desire to move from one place to another. They're actually just bouncing because of random molecular motion. So remember that they're not intentional. So why do we really care about this? Why are we studying diffusion in a biology course when it really seems to be more chemistry or physics related? One answer is because this is how molecules get around within the cell. It also helps molecules get into and out of a cell. Cells need to take in food. They also need to get rid of waste products, and that normally happens through this process of diffusion. It also helps us understand the limits of enzyme kinetics as the concentration of reactants decreases and the concentration of products increases. but that's not going to be covered in freshman biology lab. Diffusion is life. You could say that there would be no life without diffusion. All living things have certain requirements that they have to be able to satisfy in order to remain alive. We need to exchange gases, and so do microscopic single-celled creatures. The gases that we exchange are typically carbon dioxide, which is produced as a waste product of respiration, and we need to take in oxygen. We also take up water, minerals through our nutrition, through diet, and we also have to get rid of wastes. And even though we are gigantic multicellular organisms with tissues and organs that are responsible for most of these processes, our lungs for exchanging gases, all of these tasks also occur at the cellular level. That means that molecules are required to move through a cellular membrane that surrounds the cell, and that can be a bit of a challenge. Let's recall what our cell membranes actually are made out of. Our cell membranes are a phospholipid bilayer, which are made out of phospholipids, which have a very hydrophobic fatty acid interior, and then a hydrophilic polar phosphate-containing head. This lipid membrane structure makes for a really good barrier because while polar molecules that are dissolved in the aqueous environment are capable of interacting with the outside of the membrane, many of them can't pass through the lipid interior, so that makes this a selectively permeable membrane. The membrane will allow some molecules to pass through easily, mostly very small or nonpolar molecules, but others get stuck on either side of the membrane and they simply can't pass through. You can also see that there are some integral membrane proteins that are embedded within this membrane. Many of these could be used as transporters or channels that would allow other substances that normally couldn't pass directly through the membrane itself to pass through a dedicated tunnel or pore. So as I mentioned, some things can get into and out of the cell fairly easily. Small molecules like water, believe it or not, even though it is polar, and gases, dissolved gases like oxygen and carbon dioxide, can pass through the membrane fairly easily. The diffusion of water across a membrane is actually defined by another word. We call that osmosis. We often consider the diffusion of water as a separate process called osmosis, and that's a concept that you'll work on in lecture class, but not in lab this week. Let's look at an example of gas exchange as a great example of diffusion. So in this figure, we see that we have a single-celled organism that's delineated by a lipid bilayer in purple right here. And this is an oxygen-requiring aerobic organism. So oxygen, which is represented by the little black balls here, is at a higher concentration outside of the cell in the environment because it's getting actively used up on the interior of the cell. So there's always going to be an oxygen gradient where oxygen at high concentration will tend to diffuse, because of our concentration gradient, into the cell. And at the same time, carbon dioxide, which is represented by the yellow balls, is being produced inside the cell. So it's always going to be at a higher concentration while the cell is still alive, and that carbon dioxide will diffuse across the membrane to a lower concentration. This is how our cells are constantly able to bring in the oxygen needed for respiration and also get rid of the carbon dioxide waste product. Unlike gases, there are a number of substances that cannot pass through the lipid bilayer membrane. We could say that the lipid bilayer is not permeable to things such as ions. Protons, sodium, potassium, chloride ions can't pass through the membrane easily. They are very polar and they don't interact well with the hydrophobic interior of the membrane. Some small hydrophilic molecules like sugars, glucose cannot pass directly through the membrane. Macromolecules that are very large, such as huge stretches of RNA or large proteins, are too big to fit between the lipids in the membrane. So how do things like that make it across the cell membrane when cells need to be able to have access to different ions? The answer is passive transport. Molecules that can pass through the lipid membrane fairly easily are able to move through just via simple diffusion, but anything that can't make it through the membrane directly because it's a charged particle or it's too large has to find another way through, and that's usually through a membrane transporter. And in facilitated diffusion, our concentration gradients, high concentration outside and low concentration inside, can be normalized through diffusion if there's a channel or a pathway that allows the movement of that molecule to make it inside the cell. Otherwise, a concentration gradient is established and maintained where maybe these blue balls would not be able to make it into the membrane. This provides some potential energy and is going to lead the way to energy production when you start talking about respiration in lecture. Facilitated diffusion means exactly that. It's diffusion, where molecules can move from high to low concentration, that is facilitated by some other entity that's usually a channel through the membrane. There's a number of other factors that can influence the rate of diffusion, or that can influence the way that molecules make it into or out of a cell. For instance, the magnitude of that concentration difference is paramount. If there's a very small difference in concentration, then the net movement of molecules across a membrane is likely going to be very low. The permeability of the membrane to a diffusing substance can also influence the rate of diffusion. If we were to increase the length of the fatty acid tails in the membrane, making it slightly thicker, then that's going to lead to a reduction in the rate of diffusion across the membrane. Likewise, shorter fatty acids are going to lead to a thinner membrane that allows an increased rate of diffusion. We can also consider the density of the matrix through which a molecule is diffusing. If our molecules are dissolved in a very thick gelatinous substance, they're going to move through that matrix a little bit more slowly than they would if they were dissolved in water, which has a much lower viscosity. This is going to play an important role in the model system that we're using in lab this week, in which we're modeling a cell using a spongy compound called agar. It's a lot like Jell-O. And then, of course, the temperature of the solution is going to play a large role in the rate of diffusion, because we initially discussed diffusion as being the molecular bouncing of molecules against one another. As we increase the temperature, molecules move more quickly, and they bounce around a lot faster. You may have even experienced the temperature effects of diffusion if you had muscle pain. If you went out and you ran 10 miles and your back was starting to hurt, you might have purchased one of these icy hot patches to slap on your back. So how does it that the medicine that's in these icy hot patches actually get to the pain when you're just sticking it up against your skin? And the answer, of course, is diffusion, because the medication is at high concentration in the patch and low concentration at your muscles. And you can even look at the back, and there's some warnings that tell you do not bandage one of these patches tightly or use with a heating pad, which should make sense to you now if you understand how diffusion and temperature interact. If you were to bandage this and make this hot, you're going to increase the molecular motion, which is going to cause the medicine to just move more quickly into your skin, and it could get too uncomfortable too quickly. And finally, the characteristic which we're going to study in lab this week, the surface area of the membrane through which substances are diffusing will profoundly influence the rate that something's able to pass through. And we can see this in many different areas of biology. If we take plant root hairs, for instance, plant root hairs are very, very small, single-celled extensions of the epidermis of a plant root. You might recognize that plant roots are the tissue that absorbs water and nutrients from the soil. Well, if we can increase the surface area by extending some of these cells into the soil, then we have more surface through which nutrients and water can diffuse and pass into the cells and eventually make it up through the root of the plant. When we learn about respiration and mitochondria, you'll recognize that the inner mitochondrial membrane is very convoluted and it folds in on itself. This is akin to crumpling up a Wegmans plastic bag and putting it in a small area. The surface of that plastic bag still has the same surface area, but packed into a smaller space, it allows for a lot more diffusion of protons and other molecules across the membrane, which ends up serving as the chemical battery that leads to ATP production in a cell. Anywhere in your body that is responsible for absorption or excretion of gases or nutrients is a perfect place for adaptations that increase the rate of diffusion. Your intestine is a perfect example because your small intestine is a location that is responsible for the absorption of a lot of nutrients. The cells on the inside of your small intestine produce extensions or villi that increase the surface area of the small intestine to allow a greater absorption of those nutrients. People who have celiac disease, who have an immune response to the presence of the wheat protein gluten, will elicit an immune response that attacks these villi, and that leads to digestive issues. So there's a lot of places where diffusion plays a large role in biology. With this background in diffusion, now you are prepared and ready to start asking questions about how different factors influence the rate of diffusion of a substance out of a model cell. For this week in lab, you're going to need to make sure that you have at least one computer per lab pair that is capable of attaching a USB cable, because we're going to be collecting data through a USB cable. We do have a couple adapters for students who have Macintoshes or new PCs that only have a USB-C port. Also remember to bring in your phylogeny and R assignment so that can be turned in for grading. This week, you're going to be starting quiz number 3, which opens up early on Monday morning, and it's going to focus on elements of experimental design, diffusion, and dilutions, all of which are going to require a little bit of math. So make sure that you have a calculator on hand before you open your quiz. And to make sure that you are fully prepared, don't forget to watch videos number 2 and number 3 coming right up.